So we start the afternoon session with Bialek's third lecture on the statistical physics of biological systems. Thank you.
Okay, so what I want to talk about today is a problem that is actually common across many, many scales in biology. And maybe, since this is a school about ecology, I should start by talking about it at the ecological level, then I'm going to drop down actually all the way to the molecular level, which is the place I want to discuss it. And the problem is the problem of interactions in space. So as several of the lecturers have talked about, when you write down something like the Lotka-Volterra equations, you write down a term in which species I interacts with species J, you have the abundances of the species and you multiply them together. So what does this mean? It means that in some way any individual organism of species I and any individual organism of species J have sort of equal probability of interacting with each other. Now that could be directly, I mean one of them eats the other one, or it could be indirectly through one of them is consuming the resources, they're competing for resources or whatever, but it's somehow in a limit where, to use the chemist's phrase, you would say the reaction is well stirred, right? Everybody interacts with everybody else. If that's not true, then you need to think explicitly about how things vary in space. So the same problem exists at every level of organization, right? So we've talked a little bit about networks of neurons, and I said that, well, neurons are very large objects, so they can reach across long distances, but it's also not true that every neuron in your brain can interact with every other neuron in the brain. There is some structure that, that limits the spatial spread of interactions. If you keep going down, eventually you get to the molecular level, and at the molecular level, in some sense, you don't believe in any action at a distance, right? Atoms interact with each other through spaces. And so it must be that everything that happens locally, right? In order for two molecules, for example, to have a chemical reaction, the molecules actually have to touch each other. So what's surprising is that there are many situations in the molecular machinery of life where there are apparent interactions over very long distances. And that's actually the problem that I want to talk to you about today. And in order to give you a sense for that problem, and I want to show you how statistical mechanics ideas get into this, and okay, that's the plan. And I think the best thing to do is to tell you about the current problem, which is what got my colleagues and I interested in this question, and then I'll back up a little bit and give you a little background. But let me tell you about the current problem. So the current problem is in the regulation of transcription. So I worry that in the same way that many physicists who are interested in biology try to find a way that physics and biology interact while avoiding chemistry, it's possible that many people who are interested in ecology try to avoid molecular biology. So at the risk of violating that, I'm going to remind you of a few facts about molecular biology. So you all know, right, every cell in your body has, modulo the occasional mutation, has the same DNA. What makes the different cells different from one another is that different genes are read out. And that's also true about microorganisms, right? So when bacteria are in different environments, they read out and synthesize proteins from different genes which are relevant to metabolizing the nutrients in their environment or doing other lifestyle-related things that depend on where they are and what they're trying to do. And in fact, the whole idea that there is a machinery for regulating the expression of genes is something that came out of thinking about bacteria. And it's a very macroscopic, almost ecological experiment. I don't know how many of you know, do most of you know the story of the lac operon? The lac operon. Okay, so for the physicists, so look, if you're a bacterium and you live in an environment where there's sugar A, then you need to make all the enzymes that are required in order to metabolize sugar A. On the other hand, if there's sugar B, you might still be able to live, but you need a different set of enzymes in order to metabolize sugar B. So Jacques Monod did a very simple experiment. He took E. coli, which were growing on one kind of sugar, and he switched the sugar in the environment. And what he discovered was that growth paused, and then 20 minutes later, they start growing again. And so the question was, what's going on in the 20 minutes?
What's the population like?
Sorry?
20 minutes, how does it compare to
well, so
generations?
if the, it's order 1 generation. Okay. So there's a pause for a macroscopic amount of time in the life of the organism. So the question is, what are they doing for the 20 minutes? And it was their tremendous insight, Monod and Jacob, that what must be happening is that what's in that 20 minutes, the bacterium is making the new enzymes that it needs in order to metabolize the different sugar. And so there must be internally some collection of switches. And this is at a time when none of the relevant, well, DNA was identified. Okay? We knew that the enzymes were encoded in DNA, but none of the apparatus for how that information is read out, messenger RNA had not been identified, it's blah, 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 right? So we didn't know any of the apparatus, but they could understand that there had to be these switches that somehow get thrown, and then it takes time, even once you've activated the reading out of genetic information, it takes time for the proteins to be synthesized. And this played an absolutely crucial role in the development of a large part of molecular biology. And it has its origins in asking yourself, you do this very macroscopic experiment, what is it that takes, what is the cell waiting for 20 minutes before it starts growing again? Okay? Which I don't believe that the opportunity to notice things like that has been exhausted. We have a tendency to think that in this day and age, all interesting experiments are at the frontier of technology. My guess is that there are still lots of these very macroscopic things which are puzzling if you think about them, and could be the clue to something happening underneath. But we need to look carefully. And so these guys were masters of this. So let's fast forward to the point where we more or less understand what's going on in bacteria. So in bacteria, if you look at the DNA, you find some region, I'm not going to do this to scale. You find some region of the DNA that codes for some protein. For example, that enzyme that you need in order to metabolize the sugar. And in order for this information to be read out, there's a molecule which is called the RNA polymerase that has to bind to the DNA, and it walks in this direction, and it spits out behind it the messenger RNA that also codes for the protein. And as you all know, this goes to the ribosome, and out comes the protein. And there's lots of beautiful questions you could ask at any stage. And every step is regulated. Okay? But clearly, one thing you might want to regulate is the synthesis of mRNA itself. Because in some way, if you don't need this protein, there is no point in making the mRNA. That turns out not quite to be true. It might be that you want to make lots of the mRNA and inhibit its translation into protein so that at the moment when you need the protein, you can switch faster. You don't have to wait for the messenger RNA to be synthesized. And that happens. But there's also a very important part of regulation, which is to regulate this step, which is called transcription. Right? The bases of RNA and DNA are almost the same. So one says that you transcribe the information from DNA into messenger RNA, whereas in order to get to amino acids, you translate. It's the origin of the. So how do you regulate this? Well, one of the things that Monod and Jacob realized is that, and actually, this is a contribution of a theoretical physicist. What Monod and Jacob saw, of course, was that at some point, the bacterium starts to make the enzyme it needs in the new environment. And so you could say, well, it activates the synthesis of the protein. And it was Leo Szilard who pointed out to them that no, it could be that normally the synthesis of the protein is repressed. And what happens is you relieve the repression. And in fact, in the particular case they were studying, that is what happens. And so it's easy to imagine how you do that. What you do is you put a binding site in the DNA, so some particular sequence of DNA that has an affinity, another protein has an affinity for. So remember, the RNA polymerase binds to the DNA and starts to walk. In fact, if you look near the beginning of the coding region, there's a very specific sequence that you always find there, which the RNA polymerase recognizes when it first binds. So proteins can recognize sequences along the DNA. So let's get a protein that recognizes this sequence. And if there's a lot of that protein around, it will bind here. And now I've repressed transcription literally by getting in the way. So if this white protein is present, then the RNA polymerase just doesn't have any space. Okay? And so this protein, it's another protein. And in this case, it is a repressor. And more generally, these proteins are called transcription factors. Okay. Slightly more complicated would be to have a transcription factor that served as an activator of transcription. So then in that case, instead of binding here, it should bind a little further away and have the property that it being there makes it easier for the RNA polymerase to find this site. For example, maybe it also can stick to the RNA polymerase and in that sense make the binding stronger. But it shouldn't be too strong because eventually the RNA polymerase has to walk away. But certainly repression is very easy to understand as a geometrical, or as they say in chemistry, steric effect. What's wonderful is that for some class of repressors in bacteria, this picture is basically right, this cartoonish picture. So we actually know the structures of all these molecules, and that's what happens. As I said, already if you want to think about an activator, it's more complicated, but not too much more complicated. The problem is that this is a picture in bacteria. And it works because, well, this piece is actually much longer than I've drawn it, but this is sort of to scale. This part, right? That this thing really gets in the way of where the RNA polymerase has to go. The problem, as many of you may know, is that in higher organisms, I have a piece of DNA, and this is the region that codes for the protein. And just because it's a higher organism doesn't mean you don't have transcription factors. You do have transcription factors that control the reading out of the information. And they bind to the DNA and they recognize specific binding sites. So where are those binding sites? They're there. Nowhere near the place where the action happens. If you stretch this out, these distances can be 10 to the 4th base pairs. Good news, if you have a long piece of DNA and you can put binding sites anywhere along it and have them affect what's going on, then you can fit in lots of binding sites. And so regulation can be a much more complex thing. You can have binding sites not for one transcription factor, not for two transcription factors, but 10 different transcription factors. And you do, okay? And in higher organisms like us, but also like fruit flies, this is very common. Okay? And to some extent, as you go up the ladder of evolutionary complexity, it is not the set of proteins that gets, I mean, there's evolutionary biologists in the room, you guys probably know this better than I do. It's not the set of proteins that gets more complicated. It's the regulation of which cells make them and when that gets more complicated. It's also true, if you think about it, that, and this is one of the great insights of the interface between evolutionary biology and developmental biology, that in some sense, evolution can act much more quickly by changing these little pieces of DNA than by changing this one. Because in some way, if I want to do something new, I have to figure out how to make a new protein. On the other hand, if I have a fairly complete set of proteins so I can do all the functions that I want, if I start fiddling with these parts of the DNA, then I change which cell does which thing when. And so in a multicellular organism, that allows for evolution of the body plan without tinkering with the basic functions of all the individual molecules. Okay? Just do the same things, but different times, different places, and you make a different organism. And that's how, that seems to be how it worked. Okay? But there's a problem. How does the binding of a protein here influence the expression of a protein, of the synthesis of mRNA there? So, again, let's go back and think about bacteria. There are examples in bacteria where there are binding sites that are a bit farther away. And it was assumed, and eventually more or less shown, that what happens is that the DNA loops back on itself. So if I have a binding site here, I can imagine the DNA looping back, and then if the protein is there, it can also interfere with the RNA polymerase. Now, you have to figure out how to stabilize this configuration, so maybe you should have two proteins interacting with each other, a little more complicated. But the idea is that since the DNA is flexible, you can form a loop and solve this problem of what looked like action over very long distances by actually bringing the pieces of DNA together. So, is that what happens in eukaryotes? So how do you find this out? So this is where all the modern machinery of experimental machinery of molecular biology and the revolution in microscopy come together. So what you do is, for example, you genetically engineer this piece. Maybe, I don't know if I want to draw this again, but let me hand wave. Remember that what comes out here is a piece of messenger RNA. So suppose that what I put into this organism is DNA that codes for the RNA of an RNA virus. remember that in an RNA virus, the RNA is packaged in a set of proteins called the capsid, right? And those proteins bind to the RNA. So suppose I insert into the DNA the sequence that codes for those coat protein binding RNAs. Then, as this molecule, as the messenger RNA molecule starts to be spit out, if I've also genetically engineered the organism to make the viral coat, those proteins will stick to the RNA. So now you do one more step, which is to take the viral coat and fuse it with a fluorescent protein, and genetically engineer the organism to make this fluorescent version of a viral coat. So now what's going to happen is that if you're not transcribing this messenger RNA, then there'll be lots of this coat protein around, but it'll be diffused throughout the entire cell or the entire nucleus. And so you'll see a kind of diffuse background of fluorescence. But as soon as the messenger RNA starts transcribing, the coat protein will stick to the RNA, and it'll stay there. Furthermore, since you're engineering this, you can do whatever you want, and you can put 24 binding sites for the coat protein. And so what's going to happen is that very soon after this little piece of messenger RNA comes out, all of a sudden 24 molecules, 24 fluorescent molecules are all going to be in one small spot. And that will light up under the microscope. Okay? So that means that you can see whether whether there's a piece of messenger RNA coming out or not. And so you can tell when the thing lights up. So that allows you to see what's happening here. What you can then do is to engineer into these regions of the DNA binding sites for other fluorescent proteins that you've engineered into the organism. So you basically take the organism, and what you're trying to do is to instrument it, right? In the same way that when you're flying an airplane, right? There's instruments all over that are sensing various things. So what we want to do here is to genetically engineer the organism so it has lots of instruments that report on various things. In particular, they report on transcriptional activity, and then if I can just get different color fluorescent molecules to stick at different places along the DNA, then I can report on the positions of that part of the DNA. Okay? So if I look under the microscope, what I'm going to see is these spots are moving around, and then every once in a while, this thing lights up. And the question is, does it only light up when this guy gets close to this, or is there some mystery? So in the picture here, right, if I did the experiment here, I would find that if I could tag this site and monitor the messenger RNA synthesis, that I only see the messenger RNA turn on when it is in close proximity to this binding site. Or actually, in this case, it would be when it is not in close proximity because it's a repressor, but you know what I mean. Okay? And in fact, if you were a little more clever, you might want to put something here that tells you where this is, even if it's not transcribing. But okay, that's a little bit more complicated. Okay, so you get the idea? Modern molecular biology, you can play with the sequence all you want to. You can stick fluorescent markers into various places. It's not simple, but you can do it. And of course, today, you do it using CRISPR, much more precise than it used to be. Okay? Now the problem is that the scale, remember all of this is happening, for a higher organism, it's happening inside the nucleus. The entire nucleus is 5 microns across. If you buy a microscope, an ordinary microscope off the shelf, your spatial resolution is half a micron. Actually, if you buy it off the shelf, it's probably a micron. But if you work at it, it's half a micron. Being next to each other had better be better than half a micron, right? Things are supposed to touch. This protein is a few nanometers across. So there's a huge gap between the resolution of your microscope and the size of the molecules that you're thinking about. So, as many of you know, there are ways of doing better. So when you look under a microscope, if I ask you, what is your resolution? That's a question about what's the resolution you can get for an entire image. Or alternatively, if I ask you to tell the difference between one spot and two spots, can I be sure that I'm looking at two spots and not one? That's what you mean by resolution. But suppose the two spots are in different colors. Then I don't need to tell them apart. And the only question is, how accurately can I locate one spot? The spot will be a blob whose size is half a micron, because that's the resolution of the microscope. But I can locate the position of that blob to much higher accuracy. Certainly I can do 100 nanometers, and if it's bright enough, I can do 50 nanometers or 20 nanometers. Okay? And so you don't need to do super resolution microscopy in the sense of building a different microscope. You just build a very good microscope and use the fact that the question of how accurately you can localize a point is different than the question of what's the resolution of the microscope. Okay? Is that a familiar idea to most of you? Okay. It's very important. It's also true about your visual system, by the way. If you think about the diffraction through your pupil, that sets a limit to your spatial resolution, which not coincidentally is approximately the spacing between the cones in your fovea. But if I ask you, are these two lines parallel or offset, the smallest difference that you can distinguish is an order of magnitude better than that resolution. Right? Because this is about localizing points rather than telling the difference between 1 point and 2 points, right? If I ask you, is this one object or two objects, that's your resolution, and that's limited by diffraction. If I ask you about this, then if you have enough light and the signals are good enough, then you can tell much better, and you do. If you're ever in the San Francisco Bay Area and you're standing in Berkeley and you look across the bay to the Golden Gate Bridge, you will be surprised that you can see the cables of the bridge and distinguish them on a clear day. And if this was limited by your resolution, you'd not be able to do this, right? So you could actually demonstrate this. In fact, these observations go back to the 19th century, where people put things on the side of a barn and told people to walk a kilometer away and look at them, right? So it has a long history. So the question of how accurately you can localize a point is very different from the general question of resolution under the microscope, something our astronomer friends also know very well, the other way, of course. But good. So now I've genetically engineered this thing so I can measure where these points are. So the first observation is that if you're watching these things moving around, and you can do this in many different organisms, if you're watching these points move around, what you discover is that it is true that when transcription starts, the transcription factor binding sites get closer. So you declare victory. It really is true that if the transcription factor binding sites are just floating around somewhere, then they can't influence transcription. But then when they get close to the transcription binding site, something happens. So that leads you back to thinking about this cartoon from bacteria. But now you do it more carefully. And in order to do it more carefully, you cheat a little bit. You do this, for example, in a developing embryo where there's lots and lots of nuclei. And some of them are transcribing and some of them are not. So you have an ensemble from which you can choose. And you cook the egg gently so that you stop all the action. And now you have as long as you want in order to measure the positions of these spots. And what you discover is that, as far as anybody can tell, you have this complicated, actually, that's not a very good way of thinking about it. Let's try a little better. You have this complicated configuration of DNA. And remember, the DNA is not naked, right? It's got histones and there's chromatin around, and right? It's more complicated than this, but let's think about the contour that follows the DNA. And here's the start site for the promoter, as it's called. And here are the and if you start measuring these distances, you find that all these distances are on the order of 150 to 300 nanometers. And they change a little bit whether the transcription is on or off, and things move around, and they're probably not moving independently of each other and all that stuff. But nothing ever gets closer than that. So in fact, this cartoon that we inherited from bacteria is completely wrong. They have to be, in order for this to work, they have to be in proximity. But proximity is orders of magnitude away from contact. So it seems as if there's action at a distance. And action at a distance is forbidden, right? So that's the puzzle. By the way, you might worry that since all of the distances we're talking about are smaller than the resolution of the microscope, that you can't really tell. Now, I told you that, yeah, you can, because localizing a point is different than asking about the resolution, but if you're an experimentalist, you shouldn't believe arguments like that. So what the experimentalists did was they engineered two things right next to each other, which are binding sites for proteins of different colors. And so at the scale that we're talking about, you should measure a distance which is essentially zero. Right? It's just a few base pairs, so it's a nanometer or two. And in fact, what you measure, the average of the absolute value of the distance is 20 nanometers. So of course, what's happening is, sometimes you see it on one side, sometimes you see it on the other, and on average, they're about 20 nanometers apart, so that's your error. Okay? So it's not infinitely much smaller than this, but it's a lot smaller, right? So it's another order of magnitude. So these objects are 2 orders of magnitude farther away than they need to be in order to be in contact, and 1 order of magnitude farther away than the limit of your ability to resolve things. So there's definitely a problem here. Is it clear what the problem is? Good. So now let me go back actually to the same period of Monod and Jacob thinking about transcriptional regulation, what eventually was understood to be transcriptional regulation. Monod and his colleagues, Wyman and Changeux, were thinking about a different problem. And that's a problem of action at a distance at a slightly smaller scale. So let's think about now a single protein. And maybe here is the active site because it's an enzyme. So this is where, let's say, the sugar molecule comes in and gets cut so that it can eventually be eaten. It turns out that enzymes themselves are often regulated by other molecules. So for example, we know that many enzymes are regulated by putting a phosphate group at some particular site in the protein. So this is phosphorylation. It's also true that many enzymes are regulated by the binding of other small molecules. Okay. And some enzymes are regulated by the binding of many different small molecules. What's almost always true is that these events are very far away from the active site. Now, very far away in this case isn't that far. It's only 3 nanometers, but it's as far as you can get in the protein. So it's not true. So what's happening is I have this big blob of a molecule, and I push on one end of it, and something at the other end changes. And it's the longest possible distance in the molecule, right? So you have to ask, how is the information that something bound here, or that a charge, basically a charge was added over here, how does that information make it to here? Now, the protein is a more or less solid object. So maybe it's not so surprising. You push on one end, the other end moves. Well, that's not quite what you want, right? Because I don't want to just translate the whole molecule. I want to push locally on one end and have the shape at the other end change. So it's a little more subtle. But it is a solid object, so it's possible. So how should I think about this? So there's a model that is due to Monod, Wyman, and Changeux from about 1965, in which their idea was that the whole protein can switch between 2 states. So we say that the protein has a structure. So inside this blob, you know, there's an alpha helix and there's a beta sheet, another beta sheet, and then there's an alpha helix, right? This arrangement has 2 reasonably stable alternatives. In other words, the whole protein has 2 states. And in particular, for example, one of them might be a state in which the enzyme is active, and the other one is a state in which it's not active. So if you want, you could think about these 2 states flipping back and forth. And their idea was that this was actually in thermodynamic equilibrium. Which is a little weird because it's an enzyme, so you might think it's not in equilibrium, but hang on. So there's some free energy of this state and some free energy of this state, and there's a Boltzmann distribution that tells you how many you have in each state. But then the idea is that each of these states can also bind this small molecule. And one way to think about it is that if the binding of the small molecule is stronger to the active state than the inactive state, then as I increase the concentration of the small molecule, I'll shift the equilibrium. So let's suppose that in the absence of the small molecule, this state has a lower energy than this one. Well, then in that case, I'm going to be over here. But now I start to increase the concentration of the small molecule. It's going to push me down, right? But if I go to here, the free energy will be lower because the binding is stronger. And so if the active and inactive states have different affinities for the small molecule, then just by equilibrium thermodynamics, you can show that by increasing the concentration of the small molecule, I'll shift you from the inactive state to the active state. Okay? So that was the picture. Where did this picture come from? Well, actually, before this, there was the problem of cooperativity in hemoglobin. So you all know that hemoglobin is a protein that binds oxygen, right? It's in your blood. You may or may not remember that hemoglobin has 4 subunits. And the oxygen binds to each of them. And again, these distances between the place where the oxygen actually binds is very long. And we knew since the turn of the 1900s, right, since the turn of the 20th century, it was known that the binding of oxygen to hemoglobin was cooperative. And what does that mean? If you have one molecule that can bind something, right? And I ask you, what is the fraction? So let's say this is state A and this is state B. And I ask you, what is the fraction of molecules in state A as a function of the concentration of this molecule? In this picture, the answer is, it looks like this. It goes as C over C plus a constant. Okay? And you can think of that either as solving the kinetic equations for this and letting it, setting the time derivative equal to 0, or you can think about it in terms of the free energies of these two states, remembering that in order to get to this state, you have to take one molecule out of solution, so there's a term that has the chemical potential of this molecule. Okay? Or you can just do the kinetics. Whichever view you find easier, they're the same. Okay? Is this familiar to everybody, or should I do the kinetic argument? What's the derivative of A with respect to time? Well, you look for the B molecules, and you should get a rate, which is the on rate times the concentration, right? So the rate at which you go this way is proportional to how many B molecules you have and how many of the small molecules you have that can attach. On the other hand, once you're here, there's a rate to go the other way. And then everybody has to be either A plus B. So let's call the total concentration 1. Go to steady state, solve, and you get this answer. And K is K minus over K plus. Okay? First order kinetics. If you look at hemoglobin and you ask what is the fraction of sites that have oxygen bound as a function of the concentration of oxygen, you don't see this. You see this. One way to think about this is if instead of one molecule binding at a time, all 4 molecules had to bind at once, then you get the same thing, but since 4 molecules had to bind at once, you'd get C to the 4th power. Right? Because 4 molecules all have to find the same place. And this is almost that curve, it's not quite. Okay? And the reason, right, if this was, so this is almost C to the 4th over C to the 4th plus K to the 4th. And in fact, some of you may know that this is called a Hill function. So Hill was the guy who measured this in hemoglobin in 1911 or something, okay? So just, and so this is the classic example of cooperativity. Cooperative because it is as if the oxygen molecules are getting together to agree that they should all bind at the same time. But how is this possible when they're so far apart? Okay. So now that I've told you that Monod, Wyman, and Changeux had this model, you can say, oh, I can think about it that the whole complex of 4 proteins in the hemoglobin has 2 states. And in one state, the affinity for oxygen is very low. In the other state, it's high. And if you go through this argument, you'll get to something which is almost the Hill function. And I'll do that, I might do that before we're done, but it's actually not crucial. Okay. So if you go back a little further, where did this idea come from? So, and why should you believe it? So there's a beautiful story that stands behind all of this, which is in fact at the origin of one of the origin stories of the modern era of biology. So the modern era of biology, part of it is that we have this understanding that things are driven by molecules, and you can see the structure of molecules by doing, for example, X-ray crystallography. The first proteins whose structures were solved by X-ray crystallography were myoglobin and hemoglobin. This work was done by John Kendrew and Max Perutz. Perutz started his scientific life in Austria, and he set his mind on the idea of trying to use X-ray crystallography, which was so powerful for studying the structure of salts and minerals and other materials, using it to solve the structure of proteins. He had this idea while he was still in Austria before the Second World War. And one of the proteins that he was trying, so it was known that you could get proteins to crystallize. They were very soft, small crystals. And so he figured if I can get them to crystallize, maybe I can do X-ray crystallography. But of course, this was really quite far-fetched because when you solve the crystal structure of salt, right, all you're trying to figure out is where's the sodium and where's the chloride. It's not very complicated. A protein molecule, right, it's made out of, myoglobin is, if I remember correctly, 150 amino acids. A single amino acid has approximately 20 atoms. You can do the math. There's thousands of atoms whose positions you're going to have to find. So how on earth were you going to do this? So this was his vision, and it would take his life, right? Not in the sense of it being sacrificed, but it would take, over the course of his lifetime, this vision would be fulfilled. Okay, it's a beautiful story. And there he is. He's looking at his first crystals of hemoglobin. They're little tiny things, not really big enough to do X-ray diffraction yet. But, you know, if you can make small crystals, then you can probably make big crystals. So he knew he was on his way. And he was looking at them under the microscope because they were small. And apparently what happened was that the cover slip that he had over these little tiny crystals picked up a little bit. And as a result, air started to flow in. And as the air flowed in, of course, it carries oxygen. This is an oxygen-binding protein. The crystals are not so densely packed that the oxygen can't go in. This is interpretation. What he saw, just so that you can get the full mystery, what he saw is that from one side of the image to the other, the crystals started to shatter. And what he realized was that air was coming in, oxygen was finding its way into the crystals, it was binding to the hemoglobin, and when oxygen bound to hemoglobin, the structural change of the hemoglobin was big enough that it broke the crystal apart. And so he knew that if he could do X-ray diffraction on these crystals, or bigger crystals, he would not only be able to see the structure of the protein, but the changes in the structure of the protein when the protein carried out its function would be big enough to resolve. Okay. 20 years and one world war later, he had the picture. Okay. Took a long time. And I will remind you also that during that period, crude digital computers also became available, which was essential for actually solving the problem. But now, what you should imagine is that those two structures, one of which fit into the crystal without the oxygen, and then with the oxygen it doesn't fit into the crystal anymore, those are the two structures of the Monod-Weyman-Changeau picture. Right? And what that suggests is that it's not that the binding of oxygen creates this new structure, but that rather the protein was fluctuating between these two structures anyway. In the crystal, it gets trapped in one of them, right? It's mostly on one side, and then if you put it in a crystal, it gets trapped. But if it were out in solution, it would be fluctuating, and maybe 1% of the molecules would be over here, but then as you add the oxygen, you push to the other side, okay? So this is a picture of how you can understand action at a distance on the scale of a single protein. If the whole protein has two stable structures, then all you need to do is think about individual binding events biasing the free energy difference between those two structures, which is equivalent to saying that the binding constant is different in the different structures, right? So the statement that binding of the small molecule changes the bias between active and inactive is the statement that the equilibrium here is different than the equilibrium here. The statement that the binding is different in the inactive and active states is the statement that the equilibrium here is different than the equilibrium here. But this is a closed loop. So if I take the free energy difference from here to here, and I add the free energy difference to here, and then I subtract the free energy difference to get back to here, and subtract the free energy difference to get back to here, I better get 0 because I went around a closed loop. So that means that if the binding, the free energy of binding of the molecule is different to the different structures, then necessarily the act of binding will shift the equilibrium between the two structures. So that's the insight. Right? It's a very basic thermodynamic insight. Okay? So this is how most action at a distance in proteins works, as far as we know. This is the origin of cooperativity in most cases in biological molecules. There are many other examples of cooperative behavior. Okay? It is, in some sense, where microscopically, where these Hill functions come from, which many of you have probably seen in trying to make models of different systems. So maybe the answer to the problem that maybe what I should do is to think about a kind of transcriptional bubble which is where all of this action is happening. And the two states are not transcribing and transcribing. And then I have binding of the transcription factors, which shifts the equilibrium between these states. And so I have a kind of Monod-Weyman-Changeur model for this very large object, which is the whole complex of, very large complex of molecules that's involved in controlling transcription. There's only one problem. In a protein, while the oxygen molecules are very, in hemoglobin, for example, while the oxygen molecules are very far apart, there's solid protein in between. What's in the interior of this blob? Okay. So another thing that's been happening over the last decade is an appreciation that in many cases in cells, proteins and RNA molecules can form little condensed droplets inside the cell, inside the cytoplasm. And actually an example of this goes back to your high school biology class. Right? You may remember when you first saw a picture of a cell that has a nucleus, there was this little thing called the nucleolus. And people say, well, that's an organelle. And you think it's an organelle, well, that's like the mitochondrion, right? A mitochondrion is an organelle. It sits outside in the cytoplasm. It's got a membrane around it. And that membrane is actually important. It plays a crucial role in the function of the mitochondrion. Nobody actually ever told you about the membrane that was around the nucleolus, but you think, well, maybe it's just small and hard to see. So we now know that there is no membrane that surrounds the nucleolus. What's happened is that a large complex of many protein and RNA molecules have simply condensed out of solution like a drop of oil in water, right? So if I take, so you go home, you make salad dressing, right? You take oil and vinegar, you mix them up, and you get something that looks reasonably continuous. But if you wait, you know that they separate from each other. And along the way to separating, you see little droplets of the oil surrounded by the vinegar. So whenever you have a solution made out of many different kinds of molecules, one of the possibilities is that they will phase separate. And so what people have come to appreciate is that inside cells, you can also get this kind of phase separation. Now, whether it's really like oil and water, that's not completely clear yet. But you see these things, and relatively recently, in the last few years, it's been understood that, in fact, surrounding this whole transcriptional complex, there's a condensate of protein, a droplet. Let's call it a condensed droplet. And in fact, the protein that they decided to look for, which is the one they found, has a great name. It's called mediator. And it got that name because people figured out that it is somehow essential for mediating the interaction between transcription factors and the RNA polymerase. So it does something in between, but nobody knows exactly what. And what we now know is that it sits condensed in a droplet that covers this whole region where transcription is occurring. And this droplet contains order 100 of the mediator molecules. So instead of diffusing throughout the entire cytoplasm, and occasionally one molecule comes in and makes a bridge between two other molecules, what's happening is that there's a whole blob of them condensing. So, is that the solution? Is the blob, is the condensed droplet like maybe the condensed droplet has two states, like in the Manon-Weinman-Changeux model? Well, still not quite. And the reason that that's not quite the answer in all the systems that have been studied carefully, the droplets are liquid. And if you're liquid, then if you push at one end, nothing happens at the other end. Okay. So, in fact, let me now, actually, you know what? Maybe what makes the most sense, since this has been about an hour and 15 minutes. We started at 2:30, so this is an hour. But maybe, so at this point, I should do a bit of real statistical mechanics, which will be entertaining perhaps for half of you. But hopefully I can say it in a way that makes some sense, but for everybody else. But on the other hand, I've sort of set up the problem for you, and this might be a place to pause and talk about, okay, is this the right formulation of the problem, and so on. And so we should take some questions. Do you want to take a short break in questions, or do you want to just keep going?
I think it's a good idea to have a short break. Then goes to some.
Whatever you want to do, but we'll have a pause here, and we'll come back, get some questions, and if I keep 10 minutes or so at the end,
sure.
I'll do a real calculation, okay?
So we'll come back in 10 minutes. Let's first of all thank
there are quantum states, right? And then molecules have states, big molecules have states, which usually are not quantum states. They're conformational states, right? And why are they not quantum states? Well, because the system has a very large number of degrees of freedom. What you mean by the state is that there's a local minimum of the potential energy. Put the electrons in the ground. So how do I think about molecular dynamics? The way I think about this might be wrong, by the way, but this is the conventional view, and then we can discuss what goes wrong. So conventional view, right, is that you have the states of all the electrons. Now you find the ground state of the electrons and keep the electrons in the ground state as you move the molecule around, right? You move the atoms around inside the molecule. And you map out an energy as a function of the configuration of the molecule. And to first approximation, the dynamics of the molecule is classical mechanics on that potential surface because there are no high-frequency degrees of freedom left. Okay. Now, this isn't exactly right. So for example, if you watch a single hydrogen atom, the single hydrogen atom is sufficiently light and the bonds that hold it in place are sufficiently stiff that the vibrational frequencies are very high, and so its motion is quantum mechanical. And in fact, one of the things that's happened over the last 25 years is to understand that in enzymatic reactions that involve transferring a hydrogen atom, which is actually extremely common, almost always the hydrogen atom is actually tunneling rather than being thermally activated over a barrier. Okay, so the quantum classical bound, if you go back 25 years, people would have said quantum mechanics is completely irrelevant for enzymes. And at least in this corner, we now know that that's not true. So it's not that dramatic. I mean, it still just looks like an ordinary chemical reaction. But if you actually want a theory of how fast the reaction is, then you need to do quantum mechanics. And not just the quantum mechanics of the electrons, but the quantum dynamics of the motion of the atoms. So by state, we usually mean something around a local, when we're talking about molecules in states, big molecules like proteins, we mean something that surrounds a local minimum of the potential surface of the electronic ground state. And it's a local minimum, and we know that there are fluctuations around that. So by state, we mean the whole ensemble of configurations that you can reach within kT or so of that minimum. And that's a classical object, right? You've lost any notions of quantum coherence. And so the way you get from one state to the other is by thermal activation over a barrier. So quantum mechanics goes away.
Thanks. Can I make another one?
Very short. Or maybe we can
come back.
So more questions? If not, we have one, but please, more questions.
What's the saying? Things are either so clear or so unclear. Yeah.
So in the droplet condensation, what do you think is happening? I mean, this cannot be in an equilibrium phase transition, and how can that be regulated? I mean, it needs to be regulated. It cannot be happening all the time, right?
So, okay, so before we go any further, we should ask, why are there droplets? So suppose that I have a mixture of two kinds of molecules. And let me call phi is the fraction of type 1 that I have. So let's call them 1 and 2. Let me call phi the fraction of type 1. And 1 minus phi is the fraction of type 2. So what's the free energy of the system? Well, what does it mean to say that I have this fraction? So if I look in a very small volume, I might find a molecule of type 1 or type 2, and phi is basically telling me the probability. So phi is a probability like flipping a coin. It's a biased coin, right? If I go to the right scale, I basically, if I make a box that on average has 1 molecule in it, then I flip a coin, and phi is the probability that it comes up heads, and I put a molecule of type 1. So because of that randomness at the scale of single molecules, you know that one of the components of the free energy must be the entropy associated with flipping the coin. So what is that? Well, the entropy is minus phi log phi minus 1 minus phi log 1 minus phi. Right? So this is p log p for the heads, and this is p log p for tails. And I want the free energy, so I should multiply by the temperature. And if you remember, the free energy is minus the temperature times the entropy, right? When the entropy goes up, the free energy goes down. But then there's another term, right? Which is that if I look on a scale which is now big enough to have a few molecules in it, then if I have the 2 molecules together, they interact with each other. And so what's the probability that I find 2 of the same kinds of molecules in the same place? Well, there's a term which is phi times 1 minus phi. This is the probability of finding one kind, this is the probability of finding the other. And there's a constant here. Well, let me call it E0, which is some energy of interaction between the molecules. And now, if you think about it, the entropy, this term, if you plot versus phi, well, the entropy is always biggest if it's a fair coin, right? So the free energy will be lowest. So you're going to get something that looks like this. But you have to be a little careful in how you draw it because, you know, there's this funny logarithm, right? So it's actually diverging here. On the other hand, let's think about this term. If the energy here is positive, that means that the, right, that means that molecules don't like each other. So oil and water don't mix, so this energy should be positive. And you know what this looks like, but you'll notice that this term goes to 0 when phi is 0 or 1. So it looks like this. Now if you put these together, depending on what the temperature is relative to this, you get something that looks like that. So what does that say? It says that left to its own devices, the system should either have 25% of molecule of type 1 or 25% of molecule of type 2. But suppose I mix them together in some other proportion. For example, suppose I actually mix them 50-50. Well, that's terrible because that's actually the maximum possible free energy. Or sorry, not the maximum possible, but it's a local maximum of the free energy. So what's going to happen? If I take my 50-50 mixture and I break it into 2 parts, one of which is 25-75 and the other, yeah, so just to be clear, right? I'm assuming this is at 0.25 and this one is at 0.75. And this is the free energy, right? So if I take my 50-50 mixture and I rearrange the molecules so that in some parts of space it's 25-75 and in the other parts of space it's 75-25, then the free energy will go down. Okay? That's why phase separation occurs. Okay? It's a competition between the entropy, which always drives the molecules toward mixing together randomly, and the repulsion, which drives them apart. If this is the whole story, and the whole system really has a long time and can come to equilibrium, then what should happen is what happens with your salad dressing if you leave it on the counter overnight. You should have a layer of oil on the bottom and the layer of vinegar floating on top of it. Okay? That happens because they're different densities, right? But, so it just makes it easier to see. If, on the other hand, you suddenly mix things together, then what's going to happen is that you're very quickly going to try to phase separate, but that will happen locally, and so you get droplets. For the droplets to merge takes a very long time. They actually grow at the expense of one another. It's a very complicated process called coarsening, okay? And so that typically takes a long time. It is a mystery why the droplets that you see of proteins in cells seem to be stable for relatively long times at a fixed size. And so we don't know the answer to that, why you don't see the coarsening. There are different ideas, but nobody knows for sure. So it's perfectly possible for the formation of droplets to be, in some sense, a phenomenon described by equilibrium statistical mechanics. You've come to a local equilibrium, but you haven't made it all the way to the global equilibrium. By the way, it's important because the Monod-Weyman-Changeau model is also an equilibrium model, despite the fact that it's used to describe enzymes. And so in some sense, the enzyme is globally not in equilibrium because it's, you know, busily carrying out some chemical reaction that dissipates energy. But for this, it seems that for this part, you can sort of not worry about that. And I don't think it's entirely clear why.
So, more questions. We have time for maybe one more before we continue with the lecture. We have one there.
Yeah, maybe this is base or something I'm not getting right. Well,
Don't apologize.
Yeah. Well, it's really hard for me to think about you're talking about solid and liquid in respect to a protein being solid and something that is really just a bunch of molecules being liquid. And what do you exactly mean? Of course, you don't mean solid and liquid in the. Is it just an analogy or you see what I mean?
Well,
no?
So, what's a solid? A solid is an object, well, one argument about a solid is that if I push on one end, the other end moves. But another way of thinking about a solid is that if I push on it and I let go, it springs back. It's elastic. A fluid, if you push on it, your finger goes into the fluid. Now, there's a surface, if you have a droplet of fluid, then there's a surface tension which acts like it's elastic, but the fluid itself is not elastic. It flows. Solids don't flow. If I put two solids next to each other and I push, I don't get one solid, even if they're made out of the same material. You can, go home, take 2 grains of salt and push them against each other. You don't get one big grain of salt, okay? You take 2 fluid droplets, for example, put water in a plate and drop oil, right? And so you have 2 droplets. If you bring them close together, they pop and they form one bigger droplet. So that experiment has actually been done with these not with these droplets, but with other condensed droplets of proteins in cells. And that's what you see. So you push on them and they flow. You push them together. Remember, sometimes there are 2 nucleoli in the nucleus. You push those together, you get one big one. Okay? So it is literally the mechanical properties that you're worried about. Fluids, they flow, but they're viscous. Solids don't flow, they're elastic.
So in a sense, any molecule pretty much is a solid by this definition.
A single molecule is solid in this sense, yes. Now, you need to be a little bit careful in that well, so for example, suppose you have a polymer which is very, very long and has a kind of random configuration. if you grab both ends and pull, you'll feel an elasticity. Now that elasticity actually comes from entropy, not energy, so it's subtle. But in that sense, it's solid, not liquid. It's a little, one has to be a little bit careful. The crucial distinction here is about elasticity. So if it was one big molecule, then it would at least be possible to imagine. By the way, it's not obvious that this is solid in the sense that you could imagine that when you push over here, you get a local rearrangement that doesn't spread throughout the entire thing. But it's possible for it to spread because it has elasticity, right? It's not guaranteed. So for example, in Perutz's picture of how hemoglobin works, what happens is, right, the oxygen binds to the iron atom, and there's the heme group, which is the thing that gives the molecule its color. The oxygen starts out a little bit outside the plane of the heme group. When the oxygen binds, it gets pulled into the plane. As a result, the electronic states of the iron atom mix with the heme, and that changes the color, and that's why your blood is red when it's oxygenated and not when it's not oxygenated. But the iron atom is attached to an amino acid which is hanging off the end of a long alpha helix. And the alpha helix moves in a reasonably rigid way. So it acts essentially like a lever. You pull down here, and the other end moves. And so in that sense, it's solid, right?
So I think we're going to continue with the end of the lecture for,
Good. And
for a matter of time.
tell me when you want me to stop.
Yeah.
When do you want me to stop?
15 minutes.
Perfect. Okay. So how do I understand what's possible here? And the way to do this is to think about how would you know, what I did here in order to explain why there are droplets is I tried to write the free energy as a function of the way in which I distribute the molecules. So let me try to write the free energy in the Monod-Weinman-Changeux model as a function of all the things that are happening. So in the Monod-Weinman-Changeux model, I can write the free energy, and let me have a state which I'll call A, and A equals 1 is active, and A equals 0 is not. And I know that there's some free energy difference between active and inactive when I start, right? And so the idea is that there's some F0 sitting here. And actually, we know that F0 is bigger than 0 because the active state has the high energy, so mostly the thing would be inactive. Then the idea is that I have binding sites. So Ni equals 0 means that the binding site is empty. And Ni equals 1 means that it's occupied. And there's some free energy difference between being occupied. There's some, in order to occupy the binding site, I have to pull a molecule out of solution. And when I do that, I have to keep track of the chemical potential. But the chemical potential is just the logarithm of the concentration multiplied by kT. And I need some natural unit in which to measure that concentration. And then I sum over all the binding sites. Except that that's not the whole story, right? In this view, I have a variable that keeps track of whether the molecule is active or inactive, and there's some free energy difference. And I have variables that keep track of whether the sites are bound or not, and they have to keep track of every time they carry a molecule from the solution. And they might be different kinds of molecules that bind to the different sites. Actually, let's do the simple case where they're all the same kind of molecule. But then I need them coupled to each other, right? So what I'm describing here is this process and this process, but then I need to couple them together. And what I said is that the way you couple them together is that the binding energy of the molecule depends on whether you're in the active state or the inactive state. So that means that you need another term, which is that the binding energy depends on the state. Okay? So this is the Monod-Weyman-Changeux model. And for example, what you could do is to say, let me calculate, so in one view, I could say, let me calculate the probability that A is equal to 1. So remember, this is statistical mechanics. So if I want to calculate the probability of A and all the Ni, what I do is I compute 1 over Z times e to the minus the free energy, which depends on all of these things, divided by KT. And Z is the sum over all possible states, A equals 0 and 1, and all the Ni equals 0 and 1, of the Boltzmann factor. Okay? It's just equilibrium statistical mechanics. And so if I want to calculate the average of A, then what I should do is to take the probability of all of the possible configurations, multiply by A, and then do the sum. Okay? So if you do this calculation, you'll get back something that is almost the Hill function. Not quite. It's close. Okay? So that's how you take these ideas and you turn them into something formal. So now, how do I do this for my droplet? So I know that this will not have made sense to all of you. So is there some, so first of all, presumably there's some fraction of you for whom this feels a bit more like a reminder than something new. So could those of people who feel that way raise their hand? Okay. For the rest of you, is there something I could do to help? So, right, this part, right, is something that you might remember from a chemistry class, right? The probability that a molecule is in a state is e to the minus the energy divided by kT. Or alternatively, right, when you think about the equilibrium between two states, the equilibrium constant is e to the minus the free energy difference between the two states divided by kT. Okay? And the equilibrium constant is just the ratio of the probabilities of being on either side. Okay? So what this is saying is you want to use that idea, but instead of just thinking about two states, I have to think about two states of A and two states for each of the binding sites. And so altogether there's, 2 times 2 times 2, however many states I need, and I just have to sum over all of them in order to compute what I care about. Okay? Yeah?
Why are you using the probability distribution in terms of the free energy and not in terms of the energy?
So these are big molecules. And so when you talk about a state, again, as I was saying earlier, the state is some collection of possible configurations, right? It's not one particular, it doesn't correspond to the protein being in one structure. It corresponds to being in the neighborhood of some structure. And so on average, that neighborhood has an energy, but the neighborhood also has a size, which contributes an entropy. And so you really want the free energy, not the energy. Good. But you were one of the people who raised his hand at the beginning, so, okay. Okay, so hopefully this helps a little bit. So now, I need to do this for the droplet. Well, I could start with the same idea. A equals 0 means that there's no transcription. A equals 1 means that I'm transcribing. So same idea, I have a switch between two states, but the two states are now whether the RNA polymerase can transcribe or not. And I have the same idea that I have binding sites for my transcription factor. N equal 1 if it's bound, n equals 0 if it's not. And so once again, I have this term that determines the, right, that keeps track of taking the molecule out of solution. By the way, this business of keeping track of whether you're taking the molecule out of solution, maybe this is a little bit of help for the people who aren't so familiar with these things. You'll notice that the free energy goes as the log of the concentration, and it gets multiplied by kT. But then the probability goes as e to the free energy divided by kT. So that means that the kT cancel, and you get the exponential of the logarithm, which is just the concentration. So that means that when you keep track of whether the molecule is bound or not, the probability of that state gets a factor of the concentration. The higher the concentration, the higher the probability that you'll be in the bound state. Except, of course, that it would be useful if I got the sign right. Right? The higher the concentration, the lower the free energy of the bound state, right? Good. Now the problem is that I'm not allowed to write down this term, because this term would say that the binding site could directly interact with the activity at the transcription site. But they're 150 nanometers apart. They can't interact directly. Well, what I could do is to say that there's a term which keeps track of some local degrees of freedom in the droplet. So for example, in the very simplest model, the world is made out of two kinds of molecules, and phi just keeps track of the concentration of one of them. And after all, and what we said was that it would condense to give us either a high concentration or a low concentration, but of course, locally, you could have fluctuations in the concentration, so you could keep track of those variables. But in truth, we know that there isn't only one kind of molecule in the mix. These droplets have lots of different kinds of molecules in them. So let's imagine that even after the droplet condenses, there are other degrees of freedom left over. The concentration of other minor components, maybe the molecules themselves have a complicated structure, so I keep track locally of whether the molecules are in one configuration or another. I don't know. I keep track of something which are extra degrees of freedom in the droplet. Just like in a liquid, right? There's a local density, there's a local velocity of the flow. If it was a liquid crystal, there's the question of whether the molecules are aligned. I have some extra degrees of freedom. And the point is that those degrees of freedom could also interact with the transcription site, because after all, they're all embedded in the droplet. But importantly, this must be the value of the local degree of freedom at the position of the binding site, and this must be the local degree of freedom at the position of the transcription site. Okay? And then presumably, there's something complicated, which is that there's a free energy that depends on phi. And let us, for simplicity, assume that if I look at the Boltzmann distribution that's generated by this, then on average, phi is equal to 0. So we subtract the mean. Okay? So now, what do? I have too many variables, right? Not only am I keeping track of whether whether things are bound and whether the transcription is active, I have this other stuff that's happening inside the droplet. And so if I now try to compute, I can compute the probability of A, Ni, and phi, and it looks like 1 over z e to the minus f over kt. But that's more than I really want to know about. I don't really care what's going on inside the droplet. I have this intuition that when transcription factors bind, it produces activation of transcription. That's the thing I want to recover. So what do? I should do a calculation in which I only compute the probability of the things that I care about. And the way I do that is I integrate over all of the possible configurations of phi of x. Okay, I take this thing and I put it in there, and I just average over all the things I don't care about. Okay. So if you look at this calculation, what you'll see is that there's the easy terms out in front. And then I have to do one more thing, which is this term, which is the sum of gi, ni, phi of xi, and ga, a, phi of xa, all divided by kt. If you think about it, doing this integral is just averaging this over all the fluctuations that are happening inside the droplet. Right? So the way to think about this is I have the binding sites for the transcription factor, and they interact with the droplet. And I have the transcription factor, the transcription site, the promoter, and it interacts with the droplet. And now I just want to average over all the fluctuations inside the droplet. And you realize that because the drop couples to both of these things, you're going to get a term when you finish this calculation which couples them to each other. And so in fact, if the couplings are weak, right, so if the interaction isn't very strong compared to KT, right, then you can do this calculation in perturbation theory, and what you're going to get is something that looks like gi, gj, ni, nj, phi xi, phi xj, dot, dot, dot. And the really important term is you'll get a term ga, gi, a, ni, phi xi, phi xa. And so you can think of this probability as being e to the minus an effective free energy divided by kT. And that effective free energy has all the usual stuff, and then the term that I want you to focus on is this one. It's A sum on Ni, Gi, Ga, average of phi I, phi Xa. And so what this is saying is that, and it actually has a minus sign in it, and somewhere along here I probably set KT equal to 1. So, sorry. What this is saying is that when you average over the fluctuations inside the droplet, you end up coupling the activity of the promoter to the binding of the transcription factors, and the strength of the interaction is related to the correlation in fluctuations of phi across the droplet. So in order for these things to interact with each other, what has to happen is that the fluctuations inside the droplet have to be correlated over that long distance. So there's one easy way for that to happen. Suppose that the whole droplet itself had two phases. But it's a small thing, so it could flip between one phase and the other. Well, then in that case, the correlations would extend over the whole thing because, right, the whole drop would be in one phase or the other. You say, well, why would that be? Well, if the droplet is small enough, then if you tried to put half of the droplet in one phase and half of the droplet in the other phase, there'd be a huge penalty for at the surface between the two phases. So if the system were poised close to a first-order phase transition, so that the two phases coexisted, right, and the surface tension was very large compared to the free energy difference, then the whole thing would act like one big blob, and you'd be back to the Monod-Weyman-Changeux model. And so even though it isn't solid, it would act like the MWC model. The other possibility is that you're close to a second-order phase transition where you don't get the whole thing switching, but the correlations extend over long distances. So the message is that if you're close to a phase transition, then the droplet fluctuations have a long correlation length. And that gives you the appearance of action at distance. So what I hope I've been able to communicate is that by thinking carefully about the behavior, about how it's possible for information to be transmitted over these long distances, what you end up with is a picture in which it's having the droplet makes it possible for the information to be transmitted, but only if the droplet is in the right part of its phase diagram. If it's very far away from a transition, then if you do something locally, it doesn't spread throughout the entire droplet. If the droplet itself is close to a transition, then it can spread over long distances. And there's a technical question about which one of these scenarios, first order versus second order, is better, but actually at this level, it doesn't matter. So how will you be able to tell whether this is right? I think there's a very crucial test, which is that you should take these free energy differences seriously. If it's true that the free energy is related to these correlations are not independent of the distance, they just extend over long distances. But then that means that they still depend on distance. And of course, they decrease as the distance gets longer. But suppose that I'm in the configuration where the transcription factors that are activating the transcription are bound, and the transcription is on, it's active. Well, then in that case, this is a 1, this is a 1. I get minus this number. So I have in the free energy of the system minus something which gets smaller as the distance gets longer. So let's see, if the free energy is negative, but of smaller magnitude at greater distances, that corresponds to a force that's pulling the 2 points together. On the other hand, if I leave the transcription factors bound but turn off transcription, then the force goes the other way. So what that says is that if you could trap this droplet with all the binding sites, sometimes in the configuration where the sites are bound and contributing to the activation of transcription, and sometimes in the state where they're bound but they're not contributing to transcription, then there should be forces pushing and pulling the two sites away from each other. And if you work out the numbers, the strength of what those forces should produce, right, is, right, they're working against the elasticity of the whole system, right, including the elasticity of the DNA itself, right? The droplet probably doesn't have any elasticity, but the chromosome does. And the elasticity determines how much fluctuations you'll see, right? If it's very stiff, then the thermal fluctuations will be small. If it's not, it won't be. And so what you end up with is a relationship between how much the states of the different sites should push and pull the sites apart and their fluctuations, and the coefficients are exactly these coupling constants. And so the prediction is that when you do that experiment of trapping the system, you should be able to see that different combinations on average have slightly different distances. And the measurement of those distances should directly relate to the energies of the cooperativity between the different sites. So the byproduct of this picture is that the things that you can literally see under the microscope by localizing things down to nanometer resolution, which you can now do, should actually be related to the functional behavior one scale up, where you think about the free energies of how these molecules interact with each other. And that experiment almost works. So my colleague Thomas Greger, who's been doing these things, they're almost at the point of being able to declare that they can see the effect that we're talking about. So I don't know whether we're right, but I think this is an interesting problem, and I'd like to bring it back to where we started. I started by saying, you know, this issue of whether you can think of things as being well mixed or not, or whether you actually have to think about information. Things are never, it is never the case that everybody's in contact with everybody else. It doesn't work. There's not enough room in space, right? So whether you're thinking about ecology and it's the whole organisms being in contact with each other, or it's molecules being in contact with each other, you have to think about whether their spatial separation is something you need to keep track of, and do you need some extra degrees of freedom that carry the information between the entities in your model, or do they just find each other often enough that it's okay? And that's a problem in ecology, it's a problem in molecular biology. And what's cool about this problem is we know that they don't find each other directly. So somewhere there's the extra degrees of freedom that are carrying the information. I've given you one scenario for thinking about what those extra degrees of freedom are. It might be that we're wrong and it's something else, but that's the set of problems we worry about. So, thanks.
Thank you. So we are running a little bit late, but if there is a small question, we can take it. Otherwise, we go to the coffee break. Small question.
I was curious why the weak interaction approximation is what gives the result that there is coupling between the and not strong interaction.
So if the interactions are strong, I don't know how to do the calculation. Okay. So if the interactions are weak, I can do the calculation. And interactions are weak. It's an interesting fact that at the molecular scale in biology, finding an interaction which is many times kT is actually quite unusual. So it's the accumulation of many small interactions that does the trick. And so for each term, the interactions are small, but when you add them up, they could be large. And so that works.
Thank you.
So, very, very short, please.
You never mentioned the word diffusion. Is this important in timescales over there?
So this is a discussion as if the system manages to come to equilibrium. So then I don't have to worry about the timescale of anything, because I assume it was short enough that it all worked. The example, there are issues about whether you could get signaling by diffusing through the droplet. That's an interesting question. We think that that probably doesn't work, but we could be wrong. There's also the diffusion of the transcription factors to their binding sites. And there's an interesting problem there about noise in that diffusive process. Essentially, if this model is correct, then what you've done is to make transcription depend not on the arrival of molecules at a single binding site, but on an average of events that are happening over many binding sites that span a large distance, a distance which is 2 orders of magnitude larger than the size of the individual binding sites. And under those conditions, that spatial integration can suppress noise that comes from diffusion, and on a scale which would be significant for the biology. Precisely the problem that you were asking about, by the way.
Okay, so let's now take a break of 20 minutes. We'll see you here at 4:45. No, 4:45, yes. Before we head to the coffee break, a short announcement. So tomorrow the lectures are going to start at 10 instead of at 9. So I guess today you will have a heavy dinner, so there's more time for digesting it. That's it. Let's go to the coffee break. Thanks.